Welcome to the presentation on the Komodo dragon, or Varanus komodoensis, the genome and identification of innate immunity genes and clusters. This study was originally published by Van Hoyk, Prickett, Settlage et al., and is currently being presented by Patrick Hughes and Patrick McGowan under the supervision of Emily Berglund.
Study objectives. There were multiple study objectives for this study. The first and most overarching was to add to the understanding of reptilian evolution. Generically, reptilians aren't well studied or entirely well understood. As such, the study sought to help expand our knowledge base about them by studying one of the largest, actually the largest lizard alive today. One of the more focused study objectives was to sequence the genome of the entire Komodo dragon. This was done because the species is endangered and collecting as much genetic information on it is important. Additionally, it assisted in the collection of the third study objective, which was to determine what part passive immunity plays in the immune system of Komodo dragons. As passive immunity is based off of genetics, it was required that the entire genomic sequence be sequenced.
The Komodo dragon. The Komodo dragons are from the island of Komodo in the Indonesian islands. They are 3 meters long and weigh up to 150 pounds. Their max speed is 12 miles per hour, and they have a diet that consists of carrion, mammals, birds, and other invertebrates. Their lifespan is typically 30 years. As you can see, here is a picture of the Komodo dragon in the bottom right-hand corner, and in the top right corner, you see one of the many prey of the Komodo dragon, deer native to the islands.
Why the Komodo dragon and evolutionary relevance? As you might have seen from the previous videos, the Komodo dragon is an extremely large lizard, especially in comparison to the size of the most lizards, who are much smaller. Additionally, the Komodo dragon is in fact the largest lizard, and it is an extinct species of a relic population of giant lizards that used to thrive on the Indonesian islands about 3 million years ago, and longer. Furthermore, besides its size, the Komodo dragon has a very unique mouth. Inside the Komodo dragon's mouth exist at least 58 different bacterial strains, including Strep and Pastorella multicida. These strains are used to essentially poison prey when they bite into them. Furthermore, the Komodo dragon contains a gland that secretes an anticoagulant that keeps wounds inflicted by the Komodo dragon from clotting. This allows bacterial strains inflicted from the mouth to continue infecting the system and causes prey to bleed out at the same time. As such, the Komodo dragon's typical version of hunting involves biting down on an animal, then tracking it as it slowly bleeds out and dies from infection. As a result of the extremely virulent bacteria within the mouth of the Komodo dragon, it's been found that there are potent antimicrobial properties in the plasma of the Komodo dragon, which make it an interesting study topic.
Hypothesis. The hypothesis of this study is that Komodo dragons evolved a strong innate immune system to combat frequent exposure to highly infectious microbials, both through their own saliva and through the bitings of other Komodo dragons. The immune system. There are two parts of the immune system, the first being the adaptive/active immune system. First, when an infection occurs, the adaptive immune system recognizes the infection and tags infected cells or foreign bodies with antibodies. This leads to the destruction of foreign pathogens and particles within these cells and within the body as a whole. The adaptive immune system is specific to each pathogen that enters the body and must be activated with certain targets that appear within these pathogens.
Immune system. The second part of the immune system is the innate immune system. The first step in this system is infection by a pathogen or foreign body. At this point, cells within the host recognize what they perceive as foreign-looking objects. This causes an inflammation response, which triggers white blood cells to rush to the area of infection. Unlike the adaptive immune system, this system is far more generic, and the white blood cells in it are unable to attack based off of antibodies, which aren't used in this system. Instead, white blood cells attack based off of like a general sense of what seems to be foreign. Because this system is far more generic in nature and more variable, it's not based off of one bacterial strain. It is far more, it has far more variability in the antimicrobial properties. This is seen on the right in the diagram, which shows multiple different kinds of white blood cells that are used in the innate immune system to tackle the expansive host of microbial and viruses that exist in our daily environment. Unlike with humans who commonly use the adaptive immune system, the Komodo dragon's immune system is far more based off of the innate immune system, which is a common trend amongst reptiles.
Some terminology before we get into the methods of the study. First is cysteine. Cysteine is a nitrogen base necessary to form antimicrobial peptides within the body. The second is an anole. Anole is a type of lizard that is a close relative of the Komodo dragon. And finally, an ortholog. Orthologs are genes that can be traced back to a common ancestor and are similar across several species. The anoles and Komodo dragons share many ortholog genes, which we will get into further in the study.
Study design. The study can be split up into two different portions. The first was the genomic analysis of the Komodo dragon. In this portion of the study, the entire sequence of the Komodo dragon genome was analyzed and sequestered. In the second portion, the genomic analysis, or a genomic analysis, was performed on the genome that was identified and sequestered. This allowed identification of specific genes thought to be linked to the immune system to be found within the Komodo dragon genome.
Methods. There are many methods of the study, first of which was the collection of blood. Tuja, a Komodo dragon located at the St. Augustine Alligator Farm Zoological Park, was used for the collection of blood that was used in genomic analysis for the study. Second was the processing. The processing was done by white blood cell analysis. White blood cells collected from Tuja were used for both the DNA analysis and the mRNA analysis. Half of the original blood sample of white blood cells was used for genomic DNA analysis. The other half was used for mRNA analysis to form a cDNA library. White whole blood analysis was then used after another sample was taken for genomic DNA analysis. Lastly, was smearing. Smearing was used to identify what white blood cells were located in the blood and in what numbers they were found.
Methods: genomic analysis. The actual sequencing of the entire Komodo dragon genome was incredibly complicated and extensive. As such, I will only be talking about a small portion of the actual methods for it. For the genomic analysis of the entire Komodo dragon genome, PCR was performed on different portions of a leukocyte-enriched sample. This allowed different portions of DNA to be amplified in a bioanalyzer machine. Then quantitative analysis of these DNA samples was done using qPCR. This was used to form a DNA sequence library. Further steps were then taken to sequence together different libraries that were generated. By doing so, over time, through overlap of different libraries, the entire genome was sequenced.
Methods: beta-defensin and related genes. Beta-defensin genes are located in a cluster within the genome of Komodo dragons. A certain gene, known as the cathepsin B gene, or CTSB, is a gene that is located before this cluster and is present in many lizards. By identifying the cathepsin B gene in Komodo dragons, they were able to correctly predict that beta-defensin genes, which are necessary for the immune system, would be located soon after. Cross-species analysis of beta-defensin genes was also conducted. Animals have many of the same genes, although they differ in specific base pair sequences. This cross-species analysis was critical for identifying beta-defensin genes at their location and for their function. Cysteine residue also was found, which makes up the proteins that are coded for by these beta-defensin genes. Methods: beta-ovidefensin genes. Beta-ovidefensin genes are predicted to have a role in the immune system. They are found within a synthetic block being a section of genes which contains multiple clusters. They are analogous with other species that have also ovidefensin genes located within similar synthetic blocks.
Methods: cathelicidin genes. For the first part of the cathelicidin genes analysis, the entire genome of the Komodo dragon was looked at. Now, in other species such as anodids and snakes, the cathelicidin cluster in these species is located between the FASTK or FASTK and the KLHL18 genes. Assuming that similarly the cathelicidin cluster would be isolated between these two genes in the Komodo dragon, the entire Komodo dragon genome was looked or analyzed for these two genes, and the cathelicidin cluster was found between these two genes in the Komodo dragon. Further analysis on the cathelicidin cluster was done. Genes within this cluster were put into programs to determine the peptide formation based off of the genes found. These peptides were then subjected to analysis to see if they had an N-terminal Cathlin domain encoding for at least 4 cysteines, a net positive charged peptide in the C-terminal region, and a helical structure. Additionally, an antimicrobial peptide database was used to compare to the genes to see if they actually did program for known antimicrobial peptides. Results: entire genome sequencing. Overall, 1.6 gigabytes of genomic DNA was found and analyzed from the Komodo dragon. This included over 17,000 genes throughout the entire Komodo dragon genome. Furthermore, the Komodo dragon genome was found to be about 35% duplication, consisting mainly of lines and signs. Within the 17,000 genes found in the Komodo dragon genome, about 16,757 were annotated. This means that the actual locations of these genes and somewhat of their purpose and function was commented on and identified. Results: beta-defensins. Overall, 66 possible beta-defensin genes were found. Within this 66, 18 of these were identified as unique to Komodo dragons. Now, besides the large amount of beta-defensin genes found within the Komodo dragons, extreme structural diversity was also identified. Some quick terminology is cysteine residues, which are disulfide bonds between different cysteines. In the Komodo dragon, extreme variability was found in the number of cysteine residues, with large numbers of residue variance between different cysteines, some containing 5 cysteine residues, some 6, and some containing even more. Additionally, within the cysteines, or within the beta-defensins, there was variability found in the defensin domains of the beta defensins. Now, these are the actual domains of the peptides that are important in the structure and activation of peptides. So these are kind of like the activation sites or important parts of the peptides that are produced by the beta defensins. Listed below is a diagram of the actual genome of the Komodo dragon and the beta defensin cluster that was identified and used to find these beta defensin genes.
Results: ovodefensins. 6 ovodefensins were identified in the Komodo dragon cluster. VKOVOD5 was the only ovodefensive gene that was identified solely within Komodo dragons. If you look at Table 4, you'll see that it is the orthologs of both animals and snakes compared with Komodo dragons for the ovodefensin genes. Ovodefensin 5 is the only one that has no ortholog in either the animal or snake column. This is most likely due to the fact that Komodo dragons evolved this gene separately to help deal with the massive microbial environment found in their own saliva.
Results: cathelicidin genes. As stated before in the methods section, a cathelicidin gene cluster was identified. Within this gene cluster, 3 possible genes were found. Those were cathelicidin 2 barco, cathelicidin 4.1 barco, and cathelicidin 4.2 barco. The names for these genes were based off of ortholog, or their ortholog to anole lizard cathelicidin genes. Now, while there was 3 possible genes identified, there was a criteria that the peptides produced by these genes had to meet to actually be cathelicidin genes. And as you can see in the second table, Table 7, Varco cathelicidin 2 doesn't possess a C-terminal cathelicidin peptide, or at least it's not predicted based off of the virtual peptide produced. It also didn't have a net charge, which is a crucial factor in being a cathelicidin gene. It not only has to have a net charge, but it has to have a positive one. And as such, cathelicidin 2 was identified as either being dormant or non-active, or possibly not being a cathelicidin gene. However, 2 other cathelicidin genes were found.
Discussion. There were 66 beta defensive genes identified, 18 of which were unique to Komodo dragons. There were 6 potential P ovodefensin genes identified, 1 being unique to Komodo dragons. And finally, 2 cathelicidin genes were identified. These genes would have been all of these genes would have been uniquely suited to preventing microbial infection in Komodo dragons, and would have evolved so that the Komodo dragon could most definitely succeed in an environment where these microbials were present in high concentrations. Accept/decline. We accept the author's conclusions. Due to genomic similarity between Komodo dragons, animals, and snakes, the identified genes could be located and functioned to the immune system. This also presents the genetic uniqueness of some of the identified genes, which is most likely due to the fact that Komodo dragons are in an environment with a vastly different microbial infection situation than other animals. The strong innate immune system found in Komodo dragons is also due to the fact that they experience this high-intensity microbial environment on a day-to-day basis, both when infecting prey in their own saliva and when fighting other Komodo dragons.
Strengths and weaknesses of the study. There were actually a lot of strengths to this study. The first and foremost is that the study is quite ambitious and overall impressive. Anyone who's taken genetics knows that it took a lot of time and a lot of money to go out and sequence the human genome. That was a long time ago, but still, it's pretty impressive that one team was able to sequence the entire Komodo dragon genome by themselves. They did send out some of the analysis to kind of another team. However, most of the work was done by this one team. Furthermore, another study strength is that they compared almost everything to genomes found in other species, specifically closely related species, throughout their analysis of different antimicrobial genes. They constantly compared orthologs and even used those when namings genes. So this really kind of gave a strength to the study because there's not a lot of data out there on lizards, but they used what was out there to their advantage. That leads into one of the weaknesses. There's, in general, a lack of data on lizards and a lot of species that just aren't human or mammalian. As such, it's hard to make a bunch of conclusions and support them because supporting material is rare and far between. This just makes anything stated in the study difficult to understand, as well as some of the peptides and different systems used by the Komodo dragon aren't well understood, even by the researchers. There's just not a lot of research material on them, such as the case of the ovodefensins. But this shouldn't stop them. It's just a weakness that they don't have supporting material from other sources. It is also the personal opinion of the presenters that the study was weak was weakened by not having itself split into two separate articles. By including the genome sequencing and the analysis of genes, it seems as though both aspects were a little weak to present by themselves. It would have been a lot better to display one study on sequencing the entire Komodo dragon genome by itself and the methods for this, and then do another study on the analysis of specific genes with antimicrobial properties. This would have allowed more room for explanations in both categories and may have just given more overall strength to the article. A couple more weaknesses are that genes were not identified from a wild Komodo dragon. Komodo dragons in captivity have been found to have a lessened microbial environment within their mouths due to differences in food and hunting strategies. As such, it's possible if the Komodo dragon that was used for this study was a descendant of a captive Komodo dragon, that the genes in it may have been suppressed or altered compared to a Komodo dragon in the wild. So there may be differences. Additionally, as I kind of talked about this before, ovodefensins are so new that the study ends up kind of reaching for conclusions here. When trying to research ovodefensins, one of the first things that pops up is the study itself, and ovodefensins overall are kind of a new topic within the field. As such, it's hard to make any conclusions too much about their antimicrobial properties or what entirely dictates a gene as an ovodefensin, but not too much reaching was done.
Applications. There are many possible medical applications for the proteins formed by these genes located within Komodo dragons. Humans rely primarily on our adaptive immune system. So boosting our innate immune system, which the Komodo dragons have a very strong innate immune system, could prevent some infections from even forming. Another possible application is the genetic modification. By using CRISPR or other similar methods, it is possible to genetically modify organisms that with Komodo dragon genes to prevent microbial infections in the future.
Presenters' thoughts. We as the presenters came up with a few different insights or questions we'd like further explored after the study. Our first is that we would like further analysis of identified genes to be completed. Genes were identified as overdefensins, beta-defensins, cathelicidin genes within the Komodo dragon genome. However, some of these different genes are poorly understood, and the peptides they produce are also very poorly understood. And so, research on the genes themselves, how they can be altered, different mutations, and of course, the peptides they produce and how these actually interact with bacterium and provide the strong innate immune system that Komodo dragons possess would be beneficial not only in understanding the Komodo dragon, but possibly understanding humans and our innate immune system. Now, uh, speaking of the different antimicrobial genes, it's possible that some genes in the Komodo dragon were not found as parameters were established based off of other species and how identified antimicrobial genes in them had been found and where they were located. So it's possible that the Komodo dragon has other genes that didn't fit these parameters, or that antimicrobial genes in other species that weren't listed in clusters used to generate the parameters might exist that also exist in the Komodo dragon. It would also be good to study the Komodo dragon for genetic similarities and variances to other lizard species, avarians, or even other invertebrates and mammalians, as this will help us establish closeness between these species and possibly understand how their genetics work in comparison to our own. On a last note, having only one study group sequence the entire Komodo dragon genome leaves it possible that there's some error within the genome or that the entire genome may not have actually been sequenced. As such, it would just be good studying to have more groups study and confirm the Komodo dragon genome that was produced and add to it just to increase the variance as each Komodo dragon is different. So having more genomes on file could help see how the Komodo dragon genome mutates and different variances between species or within individuals in the species.